Good afternoon. Hearty welcome to our respected HOD, principal, colleagues, and my dear students. Welcome to the webinar on animal models in biological research by Dr. Mohammed Febin Farooq, former project scientist, Cleveland Clinic, US. Organized by Postgraduate and Research Department of Zoology in association with IQAC, Maharaja's College, Ernakulam. I welcome our head of the department, Dr. Rema LP, for the presidential address.
Good day and greetings to all. This is the 5th webinar conducted by Department of Zoology in association with IQAC of our college. Actually, our college with association of IQAC is taking fast strides in almost all aspects of development of an educational institution. All the departments are conducting various webinar series. We are sure that we are not, we are exploiting this COVID pandemic. For every problem, there is a solution, and here we have making use of the problem very well, and we have been conducting a series of webinars. Actually, every department, they are doing their very best to conduct such resources or making use of the resources in this era of pandemic. And speaking about our own department, we have conducted so many programs. And this is the 5th one by external resource person. Our own students have conducted awareness programs, online awareness programs. So I'm sure that Maharaja's College has never gone back. She has been actually doing very well, rather very well than the normal offline period. We could make use of time excellently well, taking classes 8:30 to 1:30, and now making use of the remaining time for such useful activities, which are very useful for our students, research scholars, as well as other fellows from external locations or outside the colleges also. So today we have got Dr. Febin, a research fellow of Cleveland Clinic, project scientist, Cleveland Clinic, United States of America. And he will be dealing with animal models in biological research. So I think this will be a very useful session for all of you, and it can utilize this session very well, especially the research scholars and those who are oriented towards research. So I'm happy to inaugurate this session for today. I welcome all of you to this session. On behalf of the Department of Zoology, I welcome all of you to this session. Thank you.
Thank you, teacher. Now I invite our IQAC coordinator, Dr. Sandoshti Varghese, for felicitation. Welcome, sir.
Respected President of the function and the head of the Department of Zoology, Dr. Rema teacher, the coordinator of the webinar, and my old friend in the Department of Zoology, Dr. Dhania Balakrishnan, respected resource person of the day, Dr. Mohammed Febin, Cleveland Clinic, USA, other faculty members of the Department of Zoology, my dear students. So when IQAC of the college decided to conduct a series of webinars over various relevant topics, Department of Zoology came with enthusiastic support for conducting all these programs for the last few days. As rightly mentioned by the head of the Department of Zoology, this is the 5th webinar program in the series, and many programs will be in the offing also. And these are all opportunities that we get to interact with the students and also with the main speakers and resource persons of this area. And to me, as coordinator of internal quality assurance cell, this is a very important opportunity to directly interact with the students of the department. As we all know, IQAC is the mandatory entity functioning in every college and in every university to promote the ideas of quality in academic activities. As we all know, quality is a very difficult concept to be understood in a very precise and unambiguous manner. Because of that, whenever we ask about the ideas of quality with anyone, that person will give his own version, his own viewpoint for quality. And that makes the problem to conduct all the activities on the basis of quality. So in Maharajas, we have attempted to define what does we mean by quality. And after a series of deliberations and after a series of discussions, not only among the faculty members, but with the students also, we have defined quality in Maharajas as transformation. Here in Maharajas, quality is transformation. And this transformation has two dimensions. The first dimension is that we have to engage in activities which will continuously enhance the theoretical knowledge, the theoretical understanding, and also its social implications. So that becomes the first dimension of quality as transformation. And the second aspect of this quality as transformation is with respect to help, enable, and empower every participant of the academic activity to take control over one's own transformation in the direction of progress. So actually that is easier said than done. So we say that we have to be the masters of our own transformation, but that is very difficult to be made as a reality. And for that purpose, in Maharajas, we have taken an approach where we will enable and empower every participant in the decision-making process taking place in the department, in the college, and in all co-curricular activities being conducted. So in that manner, by enabling every participant in the decision-making process, which is being taken not only as a democratic one, but also as a participatory one, we enable every participant in the academic activity to become one's own master over one's own transformation in the direction of progress. And with this idea, we are actually organizing every activity under the aegis of the internal quality assurance cell. And specifically with respect to the first dimension, that we have to engage continuously in that activities which will enhance the theoretical knowledge and its social connections. We are conducting this webinar series. I take this opportunity to congratulate the Department of Zoology for supporting the idea of the college in this endeavor of conducting series of webinars. With these words, I extend all the support and all the necessary words of appreciation to the Department of Zoology in this endeavor. With these words, I conclude my speech. Thank you. Thank you.
Thank you, sir, for your constant support. Now I am very happy to invite Sharmila PS, Assistant Professor, Department of Zoology, to introduce our resource person.
Thank you, Vinay. Thank you for being here in this afternoon. The resource person on today's session is Dr. Mohammed Febin Farooq, an expert in molecular neurobiology, interested in developing a career in academic teaching and research. Febin recently worked as a researcher in Cleveland Clinic, Cleveland, Ohio, before the pandemic. Dr. Farooq graduated from University of Kerala, Masters in Marine Biology, Biochemistry, and Microbiology from Dept. After qualifying thereafter, he did his PhD from University of Kerala. Sir's thesis mainly focuses on a key protein named estrogen receptor activation factor in yeast. He's an expert in fields like basic cell culture and basic molecular biology techniques. He has about three postdoc fellowships in his credit.
കേൾക്കുന്നുണ്ട്
One
ചേച്ചി
from
പറയുന്നത് കേൾക്കുന്നുണ്ട്
Department
അതിൽ
of
കേൾക്കുന്നുണ്ട്
Microbiology, University of Tennessee Health Science Center, DND Center of Disease Biology, KU Even, Zoology Clinic, Ohio. In the year 2008, he achieved international travel grant from Department of Biotechnology, Government of India. He published about five papers in high impact journals like PLOS, Molecular and Cellular Endocrinology, and Neurobiological Science. He presented papers in various conferences. Hello.
Hello.
Okay. Sorry for the interruption. He presented papers in various conferences. He had done experiments with various animal models like rat, something like geese, etc. And today, he will be sharing with us his expertise opinion on the topic animal models in research. With that, I ask you to give your full attention to Dr. Febin and help me welcoming him for the session. Dr. Febin.
So yeah, so I'm going to start my presentation here. So it's named as Animal Model in Biological Research. So let's start from the first slide. Actually, this is the poster that has been pasted in our research lab, like the including clinic, and it says that as can be thanks to the animal research, they will be able to protest for another 20.8 years longer. So it says like, so I usually like to think why is it posted there, because we all have researchers there and we all support for the animal research over there. And then it came to my mind that we have this inspection from the people who actually has strict regulations to do animal research in US every 3 months. They comes to the lab and they look at us like, are we treating the animals like very fairly? Do we really take care of them well and all these kinds of things? So this poster is there for a reason, because there are a lot of people coming to the institute. They want to see that why do we need to do animal research, and for them it's hard to understand why really animal research is needed. And going to the next slide, actually this is Stephen Hawkins. When he started writing a book on a brief history of time, his a publisher asked him that like if your book really want to be famous, then you have to actually reduce the number of mathematical equations to almost nil. So he says that like if you like for one equation, yeah, the popularity of your book will be reduced by around like 10%. So if you include like the 10 equations in your book will go nowhere. And remember that he's going to write a book on theoretical physics about time, and he has this constraint, but still he wrote this book, a best-selling book, with just one equation in the entire book, and that equation is equal to mc squared. I took that advice from the editor that in my presentation I'm going to be as simple as possible, so it's not going to be like very technical in my presentation. It's like my experience as a postdoc, how animal researchers are done. What we learn in the textbook is quite different from what we do when we actually do the research, things like that. So it's going to be very basic really, and in the first part, and then we will talk about some of the model organisms I actually worked with. So continuing with the next slides, well, I give you a simple definition. You can actually Google it from internet. What is an animal model? And it says that an animal with a disease, either the same as or like a disease in the human. So it's like either you have a same disease in an animal, or it's somewhat similar to a disease in the human. That is what we call an animal model. Animal models are used to study the development and progression of the diseases and to test new treatment before they are given to humans. That's a definition they actually say for animal models. It's actually true that because we try to model a lot of diseases in these animals, but exactly are they equivalent to the human disease that sometimes it's yes, most of the times it's no. So when we deal with animal models, there are a lot of things that we have to be taken care of. So when I was actually planning to put a title for this talk, I was initially thinking to give a title like whether do I need to give it like my what is an animal models in biological research, and like a questions possible caveats. Then I thought maybe it's kind of like a little bit of negativity. So then I thought maybe I can actually put another title like trials and tribulations in animal research. Then I still thought maybe it's again sounds like a little bit of negative. Then I came with the title just a plain one, like animal model in biological research. But the focus of my talk is going to be like give a simple explanations of different animal model that you use and different criteria set on what basis we select the model, and give you an experience of working in my experience of what do we expect when we get when we work in with an animal model. So you can actually have animals with transplanted human cancer cells or other tissues called xenograft models also, and then you can actually use a normal animal to study the normal physiology of an animal. So for example, the typical example is a sleep study that we do using zebrafish and even in Drosophila. So this is a replication site. I'm going to explain it to you again. This So you have this animals used in the biological study. The first animal that comes in our mind is the mouse, the most favorite laboratory animals. And mouse genes are about 80% orthologous to the human gene. So, and it is similar anatomical, cellular, and molecular characteristic to humans. So we all use a lot of models using the mouse. You can see there's various breeds of mouse available for research, and then this is how a typical mouse facility will look like. Like you can actually put somewhere on like in a very confined places in an animal house. So it's very handy in a way. And then other comes the rat, also the favorite laboratory animals, and most rat genomes are shared to the human genes. And it also shares similar anatomical, cellular, and molecular characteristics of to humans. And this is a typical rat cage, and is slightly bigger than what we have for a mouse cage. The size matters in the case if you are doing research in today's era, because everything it matters with money, because larger cage means you need more room, and more room means more money from your grants. This is another favorite guy, the fruit fly. It's very favorite in a sense that it has a very short generation times, like around 12 days time, and a very well understood phenotypical levels, and has simple genome. It is very well understood in the phenotypical level, like it is used in developmental studies, that if you actually have a pattern change in the wings or in the eyes, you can actually trace back to the molecular pathway that actually is influencing that development. So 60% of the gene involved in the human genetic diseases and on some cancers belongs to this guy, and used in developmental studies, cognitive behavior, and even in learning and memories. So in my talk, I'm going to actually give you examples about the work done in Drosophila, and also in rat, and also in mouse. But there are other animal models also, like the zebrafish. This is an excellent model for the developmental and disease studies. And basically, this is because that the larva is transparent, as you can see here, and it is genetically well characterized. That you can see actually the larva labeled with fluorescent proteins. So it's very easy to visualize the changes that is happening during the development of these model animals. And then you have like rabbits, mainly used to do the polyclonal antibody development, pregnancy model, and also immunological research. And you have the frog here that is used for evolutionary and ecological studies, embryonic development, metamorphosis, regeneration, physiology, and all these kinds. And then you have various other kinds of animals, like guinea pig, hamsters, chinchilla, gerbils, and C. elegans. All these kinds of animals are used in various researches. Now, how do we select an animal model? There are two broad classifications of models. One is like an analog, based on analog, and then based on homology. Analog means that similar structure and says that similar functions. Is in case here is actually the penguin, and the fish both has fins that they both use for swimming, and the web feet for also used for swimming. So that means they have similar structure, and we assume that they have the similar functions. Another broad class is based on homology, like similar evolutionary precursor means having similar functions. We mainly actually we shift in both directions, like from analog to homology, whenever we choose the disease model organism and explaining the physiology in humans. Now, how do we select? And again, there are four main categories of animal models. First one is like induced or experimental model. And then you have spontaneous or natural models, and then negative or non-reactive models, and orphan models. But if you explain what is an induced or experimental model, means an attempt to reproduce conditions found in the original species. That is like if you have a disease in humans, we try to actually induce that disease parameters in the model organism. That is the most research is going on. That's the kind of model that we use quite often. And then the spontaneous and the natural, similar to the condition in the original species. You once in a while actually comes in touch with the spontaneous models or natural models that is naturally available. You don't have to do anything, and you can actually study that models too. And the negative on the non-reactive models is kind of like a positive control for this. They are the normal counterpart of a disease model. I would say, like if you have If you have a transgenic mouse, you will have some litter mates actually that are perfectly normal wild type, and then you have certain disease-causing organisms. So these negative on the non-reactive models, that's a wild type, will be a perfect control for these this disease model. Then you have the orphan models in animal diseases in which no human or animal counterpart is known. It's just like a random disease that we may or may not be interested in these kinds of models. So, as I said, the majority of the research is actually based on induced or experimental models. So we have to actually create a disease like phenotype in an animal before we actually study it. So, and finally, the selection is based on the following parameters. Actually, this is actually what we considered the last to select an organism. First one is appropriateness of an animal. As I said, like you have to actually relate it to something to the humans, because at the end of the day, we don't want to treat tumors just in mouse. We have to actually carry that research to the human counterpart. Otherwise, it's kind of like, I won't say that it's waste, but the funding agencies really want to know what we're going to do next after this. And then transferability of the information, that once you get some information, you have to actually transfer it to the next level, right? Like it shouldn't be like in an isolated point that's at a dead end. It should usually have to be taken to the next level, and that rather than next level, it has to be actually, for example, if you do some researches in mouse, it has to be a way to transfer it to the next complex organism, rat, or the higher parameters. And then the genetic uniformity of an organism. When you select actually an organism, the genetic uniformity is really very important. For example, like you have different strains of mouse, and when you actually do an experiment, you have to be very careful that you have to have the basic background very stable. Otherwise, the results will be very much varied with different backgrounds. So if you are using a black 6 animal, that is a black mouse, you always have to stick on to that, and that uniformity is very important. So that's why we normally nowadays use all these lab animals. For even in some labs that we just concentrate on the same vendor that we get the animal from, and we make sure that it is from the same gene pool. The background knowledge of a biological property of an organism is also very important, because we need to have some information about these animals. That's it shouldn't be totally new or naive for us, so that we can actually go back and see what's really happening and what is available in the literature. And the cost and availability, this is a very important factor. As I said, like as the animal size increases, as the complexity increases, you have to have larger cage spaces, you have to pay more for these animal facilities, and that will go from our grant, so we will have less money to do the actual research. And then the availability, that is, it's like nowadays it's like mouse and rats are very common, and you can actually get as much as you want. But the main availability factor comes when you actually have some strict protocols in place, that you cannot use like as many animals as you want. You have to give justification for using additional rats or mice. So we have to be very careful in selecting those animals. And then generalizability of the results, that I said, like you have to have a result, and then it should be like applicable to general area, that you have a mouse result, and we can actually see if it's the same with the rat system or Drosophila or even the primate system. We actually think it's going to be, I would say, it's going to be uniform in all these higher mammals, but sometimes it's also not the case, because, for example, the result that we get in Drosophila may not be very much applicable to the mouse. And we say, yeah, it's because flies are flies and mouse are mouse. And then easy of an adaptability to experimental manipulation. This is something that we actually consider much value, because we usually use a lot of mouse models, because mouse are vulnerable to so many genetic gene manipulations. So you can make lots of transgenic mouse and all these kinds. That's why one of the reasons why mouse became so popular in animal models. But nowadays you can do the same kind of things with rat too. Now, ecological consequences. So ecological consequences, because nowadays everything is actually from laboratory vendors, so we are not getting it much from like attention to ecological aspects of things, because they are bred in captivity just to do the research, so it doesn't matter much nowadays. Now, ethical implication is something that is very important to consider, because I have seen many labs in US closed. Initially, it was working with dogs. Now, the American people are actually very much close to their dogs, so they don't allow any dog research anymore. So it is absolutely essential. And also now we have to use very less number of animals to stop cruelty in these animals. So it's kind of like we have to carefully, as I said, we have to carefully design the experiment. So we have to give a lot of explanation why we are using this much animals. So ethical implication is really something that we have to take care of. One of the reasons is that we cannot actually go in higher primate ranges if it is absolutely necessary, unless we have to do that. Otherwise, it's like they will ask you to go back to the lower mammals. But that's one of the ethical implications that comes. So how do we generate an animal model? Traditionally, it's usually used in incompetent and immunodeficient mice. So we actually inject it with the synergic and xenograft tumors transplanted subcutaneously or topically. So you have the tumor models. Once if you have this immunocompromised mice, you can just surgically integrate it into the tissue and you can study it. And nowadays, we use more as transgenic insertion of foreign DNAs, like transgenic mice. Its definition is like insertion of foreign DNAs from other species. So now, types of transgenic animals. I'm not going to go more technically into this. It's going to be like more like the explanation for itself. Like we have two types of transgenic animals, that is loss of function, and we have the gain of function. These are the two types of transgenic animals we'll be dealing with. And one, in case of loss of function, you have the constitutive knockout, and additionally you have the conditional knockout. The difference is that the constitutive knockout is it's knocked out from this entire start of the life. So certain genes you cannot, housekeeping genes you cannot actually knock down from it, because it will be lethal embryonically. Then you have to move to conditional knockouts, in which you can actually knock out the gene at a later phase of its development. And then you have the gain of function again. It's overexpression of certain genes. The other one is the complete knockdown of the gene, and the gain of function is you have like it's increased expression of certain genes. It's constitutive random insertion model you have. Then you don't have any control, that you just insert the DNA into this, it goes into the genome and it starts expressing. And then you have the knock-in pharmacy locus model, in which you have some control, in which you can actually guide the foreign DNA to, so that you know the amount it is expressed and how it's expressed. Then you have the conditional knock-in models, in which you can actually turn on the gene at certain point of time. I will give you an example in the later part of my talk and how that works. And then you have the reporter knock-in models, in which you have reported genes as a so that you can actually look into the area where in which the protein is. One typical example is that the rats with the calcium channels, in which you can actually see the firing of neurons. Now, these are all germline genetically engineered mouse models, because these all changes are happening in their germline. So it is like uniform throughout the animal. So whichever cell you take, there will be some effect of these things. But there are non-germline genetically engineered models too. And they are produced by generating chimeric or transplantation models. In research, in cancer research, actually, these kinds of models are preferable, because they are more close to the human disease scenario, because in humans, you have certain patches of cancers in certain regions. It's not in the entire tissues, in the entire cell. So, and then there are another alternative DNA modification techniques that you can use, that is also not germline genetically engineered models. An example is like using transposons and jumping genes based on insertion mutagenesis. That is, example is like Sleeping Beauty, piggyback kind of things. And then you have RNA interferences. You can actually choose to knock down a gene at a later phase of its development. And then you have the engineered nucleases like talent-mediated gene editing, and editing using zinc finger nuclease, and now the much celebrated CRISPR-Cas system that you can actually knock down the gene in any animals, including the rat or so other animal models that we can actually come across is like induced by certain chemicals or irradiation. For example, irradiation models, we have like sublethal dose of gamma irradiation. I will talk about that. More models also in the later part of my talk. And then you have the chemical induced models. A typical example is cyanic acid induced model for epilepsy. And then the PTZ, that is pentanyltetrazole, that is a GABA inhibitor, also that is an example of epileptic model. Then you have the animal shock model of shock model, ischemic stroke, that you literally tie the blood vessels with ties so that it actually causes a stroke-like phenomena in the brain. And then you have traumatic brain injury models. You use a percussion devices to give an impact in the rat or the mouse head. So these are other types of animal. In no way these are it's not the entire animal models I'm talking. I've just picked a few so that it's not overwhelming. So now let's look into some animal models that I personally come across. So first one, I'm going to talk about an animal model that in this part you usually have like two different models that we use for this study. One is the rat model, and the mouse model, and the other one is the Drosophila model. So this is going to be like a little bit technical. So you have a gene UBE3 that expresses a protein like E6AP. So we wanted to know what this does in two disease models, that is autism and Angelman syndrome, and also we wanted to check how this proteomic profiling happens in Drosophila. So we use two different model organisms. So one is the mouse and the other one is a rat, and then I will give you an example how we actually did the experiment first. So we have two disease models also, like autism and Angelman syndrome. So let's talk about how UBE3 is linked to autism and Angelman syndrome. So you have the chromosome 15, Q11 to Q13. This is a highly volatile region in this chromosome because it undergoes a lot of transformation, deletion, duplication, all kinds of things that happens in this. So I would like to give an explanation of what Angelman syndrome and autism is. Angelman syndrome is actually a maternal deletion of this region, that is in chromosome 15, regions between 11 and Q3. It can cause by the deletion of this region, and it can also cause by paternal uniparental disomy. And it can also cause by imprinting defects, and it can also cause by mutation affecting in these regions. So it is a loss of function disease. Here we have in Angelman syndrome. Autism, it's the other way actually. It's a maternal duplication. So here you have a duplication of this region, and you have an increased dose of genes that is expressed in these regions. So you can see there are multiple genes in this, and UBE3, a gene which causes the causative factor of disease, is also present in the same region. So you have a link between this disease and these syndromes. So if you actually look on the way in which actually the UBE3 expression pattern is governed, you have a normal range of UBE3 expression somewhere in the middle. And then you have Angelman syndrome, that is total loss of function of UBE3 gene that goes all the way down. So that means there is no UBE3 expression. And then Angelman syndrome, that is paternal uniparental disomy. That means that you have only one gene from one of your parents who still have a little bit of expression, but it's not enough in the normal range. And then you have the Prader-Willi syndrome, that is maternal duplication in this region. So you have increased expression of UBE3, and then you have autism, that is interstitial duplications. So they have increased level of UBE3 in this, and then you have autism, that is idiocentric duplications, and so it's a little bit more duplicated in these genomic regions. So you have more of UBE3 expression. So this region is called, if you come in this region, you have this is called autism spectrum disorder. So this is a spectrum of disorders. You have to characterize which one it is based on the level of UBE3 expression. Now, coming to the function of this is a protein, that is the gene UBE3 is expressed, a protein called E6AP. So it is a E3 ubiquitin ligases. It targets protein for degradation and cellular trafficking and phosphorylation, site-specific cleavage and all this. And it also acts as a cofactor for steroid hormone receptors. Now, there is an important finding that the Drosophila ortholog, now it comes like, as you know, this ortholog gene, UBE3A, called the DUB3, can regulate monoamine synthesis indirectly through the transcription of a co-activation of GTP cyclohydrolase 1. What is the importance of this factor? Because these spectrum disorder people has neurological disorders that is assigned to neurochemistry. So does UBE3A regulate monoamine changes in autism syndrome and ASD mouse model? That's what we actually looked into these animal models. So we used 3 types of animal models. That is a maternal deletion, that is knockout of exon 2 of UB3. So that is maternally inherited example. So it doesn't have any UB3 expressions in these animals. Then you have the maternal duplication, that is 15q is increased, so the expression is increased in these animals. And paternal duplication, this is, so if in cases, I forgot to mention you one thing, this gene is imprinted in the sense that it is maternally imprinted. So you get this expression only if you inherit the gene from your mother. If you get the gene from your father, it is silent. So if you have a paternal duplication, it normally doesn't affect much in this case. So this is a Western blot analysis that to show that you have a knockout animal. If you can see here, there is no UB3 expression. We used both the region, that is hippocampus and the cerebellum. In the cerebellum, there is nothing. In hippocampus, also you see no expression of UB3 in the maternal deletion. Then you have the maternal duplications here. You have increased expression, and the paternal duplication, it is not that much. This is the quantification of this graph. We can actually see there is almost null expression in the maternal deletion, and in maternal duplication you have increased expression, and in paternal duplication is almost close to the wild type. So here is the immunohistochemistry that you see in the cerebellum. The Purkinje cells are actually labeled with E6AP antibody. You can see that these cells are all labeled. In maternal heterozygous, there is absolutely nothing there. Maternal duplication, you have increased expression, and paternal duplication, you have close to somewhat like a wild type. So what we actually see in these results, that you have like serotonin expressions is like you have increased serotonin expressions in maternal deletions in different regions of the brain. I'll quickly go through these results. And you have dopamine, actually, it goes in maternal duplications too. And then you have norepinephrine also expressed in maternal duplications too. So what I'm actually telling here is that you expect an exactly opposite results for these animals, because here the animal model is exactly the opposite. Like you have a knockout animal, that is maternal deletion, and then you have a maternal duplication of maternal duplication. But the results are not always the same. You have like you have in some regions the monoamines goes up, and then the other region it goes down. So you cannot always expect a reverse. you cannot always accept a up down kind of like thing happening in these animals. Now we also did some home cage licking analysis for in these mouse models. Here you can actually see this is a highly coordinated activity, motor activity that is needed by the mouse. So how we did is actually you have this is how a mouse licks its bottle. So to drink water. So you have a wired platform here. This will be having a small current in it, and then you have this. The bottle cap is also will be connected to some kind the small circuit. When the mouse comes and licks the water, the circuit will be complete. So the signals will be actually recorded in the computer. So what we are seeing here is a highly coordinated cerebellar function that is also involved in these involved with the UB3. You can see actually in normal the mean lick interval is very much different in when you have the maternal deletions. So it goes high compared to the wild type activities. So you expect the same kind of result when you have you expect the opposite kind of result when you have the duplication, but that's not the case here. Actually, it's a deletion model. You see an increase in the mean lick interval, but you see in wild type also you have the same kind of increased lick intervals in the duplication. So what it says here is that you cannot actually expect an opposite effect in deletion and duplication models. And why? There could be multiple reasons for that. In this case here, you see actually this is the genes that are duplicated in the autism mouse model. You can see just not UB3A, but tons of other genes. They can actually have an influence. So we analyze the effect of other genes. You can actually see whether the other genes are imprinted or they are expressed in different ways. Yes. So here you can see in red, all the red bars goes up in most of the animals. That means that for most of the genes, so that means that in paternal duplicated animals, you see an increase in certain genes that can also have an effect on these animal phenotypes. So what I'm saying is that the models that you use in biology is not as simple as we usually think. So, but that's a good thing because now you start up with a single gene like UB3, and now you end up with like multiple genes to work with. So it's always you solve one problem, you get a 10 more ones. The next, let's talk about another system model that we use. The model system that we use that is a algorithmic profiling of the same gene. This is actually in Drosophila. So in Drosophila, you have this the Gal4UA system that you can actually use to increase the expression of certain genes. So here it's kind of like a conditional knock-in model because this works under the promoter of a Hitchcock promoter. So you grow the transgenic flies in normal temperature, they don't actually express you a transgenic proteins, but once you increase the temperature slightly, the Hitchcock protein promoters activates and you get a lot of your recombinant genes. Here we use actually 4 kinds of constructions to deal with the study. One is the loss of function UB3, in which the protein is expressed, but it doesn't have a kind of like an ability to function properly. Then you have the human UB3A, that is a human analog that we made it and transformed it to the Drosophila. Then you have the fly UB3A, that is the fly protein itself. And then we have the mutant UB3A, that is it doesn't have the ability, it doesn't have the cofactor ability that it acts. So we have 4 different scenarios here to see the differential protein expression. How we did it? So we, as I said, these genes has been transformed, get transformed to the Drosophila, and once we increase the temperature in which they live, they start expressing the protein. And once they have the protein, you take up the head, separate the protein based on the pH by isometric focusing. This is actually for the first part of this. and then you cut down the strips and you separate it on a normal STS page, and you get somewhat like a differential protein, a kind of like 2D, but it's not exactly the 2D, but it's done in separate fashion. So you can actually isolate, for example, I have shown a strip here, you can see the differentially expressed proteins here, and you cut the band out and send it for MALDI-TOF analysis to identify the protein. So this is how we use the other model organism in Drosophila to study the UB3-related genes. So the results are like we identified nearly 49 proteins where differentially expressed genes that is involved in ATP synthesis mechanism, actin cytoskeleton integrity, and some involved in neural development and malfunction. And we pick up a few, and later we went for the downstream analysis. One is the APS15 and the ATPS5. So you can actually see here the like validation of the result in which you express the UBH protein, its limiting state of the protein is changed, but when you have like a non-reactive protein, that it doesn't change much. So you have like the polyubiquitin tail for these proteins. It is an example of ATP alpha. So you can actually see when the components for the ubiquitination is complete, you see the streak along the gel. So these are additional validation of our results that we say that these proteins are actually UB3 mediated. Now I'm going to go for another mouse. So I'm going to go for another mouse model. This is a neural tube defect, DYT1 dystonia mouse model. And there is something peculiar about this model, like so dystonia is also a severely debilitating neurological disorder, and it is actually characterized by repetitive contractions of the muscle with painful twitching movement and abdominal posture. It's very hard to see the patients with dystonia because they will be like twisted and we feel their pain actually when we see them. And it's caused by the deletion of the glutamic acid of the tyrosine A protein. So between the 302 and 30A. So this is also called as tyrosine 1A dystonia. So this is a AAA plus protein, and it's mainly actin recycle transport and protein degradation. So the tyrosine A activity is directed to the nuclear envelope. You can actually see that this is a wild type in which the normal nuclear membrane. Here it's actually the double knockout in which you can see blebs, abnormal nuclear membranes that's happening in these rats. So the loss of function of TOB1A causes a phenotype called exencephaly. That is like the brain grows out of the cavity, the cranial cavity, and it has also the curly tails also. So an important factor that you have to see here is the table. You have the wild type that is 0%, there is no exencephaly, and then you have the total knockout that is like 7 out of 18 is exencephaly. That means it's not 100%. This loss of tyrosine A is not causing this disease in 100% of these mouse models. This is what we sometimes call as incomplete penetrance. So this is also what we have to expect when we work with our mouse model, because when you cut down a gene or protein, it doesn't usually mean that you always get a phenotype that you expect. So now the table, you can actually see that this is another way of actually explaining the result. This is the rescuing method. Here you have another protein called sun protein that is also interact with the tyrosine protein. So if you actually have a knockout for double knockout for tyrosine and sun, this is like a partial knockout. You have rescued the phenotype a bit. And when you have the double knockout for completely, if you knock out tyrosine and sun protein, you completely rescue the genome, the phenotype that is see, you get no accidentally in these animals. So this is how sometimes the animal model works. You sometimes get like incomplete penetrance, just like what we have right now with the COVID-19 pandemic, right? So we don't know what's going to happen if I get infected. Will I have any symptoms, or I will be like have mild symptoms, or I will be hospitalized? We never know, right? So this is exactly what happens with these kinds of animal models. Now, we can also, I'm going to talk about another disease model. It is actually also another dystonia. It's called Micronus Dystonia. This one has actually the same twisted posture, but it also has myoclonic jerks actually. So it is characterized by very rapid muscle contractions. So it's like additional phenotype in the humans. They also have dystonia, and then you have this myoclonus jerk. So usually in humans, this disease is caused by the mutation of SGCE. It's a sarcoglycan, epsilon sarcoglycan. This is they have the paternal mode of inheritance due to imprinting in maternal. So if you have a disease gene that you inherit from your father, you most it's autosomal dominant, so most probably you will have this disease. So what does the sarcoglycan does? I'm going to give you a brief idea of what the protein does. It is a 45 kDa single. It's a membrane protein. It's a scaffold protein to be short. Actually, this is a part of a huge complex because this complex is very well studied in the muscles, but not much in the brain. So you have a very big complex, and you have 4 subunits of these sarcoglycans, different ones. You have alpha, beta, gamma, and epsilon, and then you have all kinds of membrane proteins along with distant complex. So in all of the tissues, so epsilon sarcoglycan is unique in the brain. So they are found only in the brain and also in very early stages of development. And the characteristic feature of these sarcoglycans are, if you actually knock out one sarcoglycan, the stability of this complex will be compromised, and you will have less expression of all these other sarcoglycans. So these are the different type of complexes that you see in different tissues. For example, in skeletal muscles, you have alpha, beta, gamma, delta, and cardiac muscles also. In smooth muscles, you have like epsilon, beta, gamma, and delta complexes. So different tissues has different complexes, different complex of the sarcoglycan. So no, we went in characterizing these DGC complexes in our SGCE, that is sarcoglycan epsilon sarcoglycan nocturnal lysis. This experiment went in a very interesting fashion. So see, for example, if we have 2 results here, one complementing the other. So this is a real-time PCR result, and this is an RNA-seq result that is done for the same gene, actually, set of genes. You can see this is the basal expression of this protein, and all goes up is increased a bit, and all goes down is reduced a bit. So if this one is SGCE, that is epsilon sarcoglycan, this has like around 14-fold reduction in RNA-seq results, and like close to like 7-fold reduction in real-time PCR. This says that the animal is actually a knockout because they don't have any epsilon sarcoglycan gene, and this is the gene result. So they don't express any mRNA. So far good. But now when we then characterize the dystrophin complexes in the brain, nerve, and lungs, here you can actually see dystrophin, wild type brain as whole brain, and cerebellum, you see the bands, it's there. And in sciatic also, it's slightly reduced. In the sarcoglycans, when we went for looking for the beta, delta, and epsilon, they are also there. This is very interesting because we knocked out the epsilon sarcoglycan, but you see the band there in these animals. So if you go back to the slide, this is a real-time result in which this gene is reduced or down regulated to 8-fold, and another one is 14-fold. And when you actually go to the protein, you actually see that thing. So this is the same with the different, but in the sciatic nerves is actually not there. And in lungs also you see no epsilon cycloglycan expression, but in the brain you still see the epsilon cycloglycan expression. So what does it mean? It means that the brain express the epsilon cycloglycan? No, because you see the mRNA level is almost like nil here. But why is it showing the band here in the brain? So this is where we have to keep in mind that we cannot always trust all the reagents that we get from many of the vendors. So sometimes we have to make our own tools to do the research. This doesn't say that the protein is there. It says only the antibody that we're using to detect these proteins are not good to detect the proteins from the brain, because there is no epsilon sarcoglycan, but you see the band there. We actually, when I was actually doing these experiments, we tried almost 90% of the antibodies that was available commercially at that time. Then, yeah, we developed our own reagents for our studies. For example, we use the nanobodies here. So conventional antibodies are very big. Then we have the single chain antibodies. It's slightly smaller, but not as small. Then we have the nanobody that's very small. So here we have the nanobody generations. You used it we purify the protein, inject it like in the llama, and then you generate the antibody. So this is actually the total protein samples that you do with the brain isolate, and you actually have the knockout brain. This is the traditional antibody, and here you actually see when you use the nanobodies, your protein is gone. So what it says is like, even though sometimes when our organism doesn't like a good choice, you can always actually look for another reagent studies to give more valuable results. So now, the last part of my talk actually is going to be something, another aspect is like, we take complex model organisms and then split it into simple models. Here, this is an in vitro culture model for irradiated rat models of epilepsy. So this is the recent work that I was actually doing, and this is the work is still under the process of publication. So what we did here is we took a complex models, that is an irradiated rat model, and we isolated the cell from the brain cells from it, and then we made it into a simpler format. I will give you an example of how it works. So what is an irradiated model of cortical dysplasia? So when the mother is pregnant on day 17, we irradiate the mother with gamma irradiation. So the pups that are born is called the irradiated model for cortical dysplasia. This is called as XRD. So what is cortical dysplasia? This is cortical dysplasia is a precursor factor for epilepsy, one of the major contributing factor for epilepsy in US. So this model is actually a very good model to work with, because it almost duplicate most of the factors that you see in the human cortical dysplasia. Here you can see control and irradiated brain. Of course, the brain is smaller than the control one. The major hallmark of the dysplasia is that it has a disorganized cortical architecture. Here you can see well-organized neuronal cells, but here the organization is completely lost, and you will have neuronal clustering in these animals, and then the cerebral cortex is thin, and you have a typical feature called hippocampal burst here. So we don't know exactly what it does, but this is a very characteristic hallmark of this dysplasia. So they have a low threshold for PTC for the generation of seizures. So what happens is like the cortical dysplasia lays a framework for epilepsy, and they develop epilepsy spontaneously in the later part of their life. So this work has a two-hit hypothesis of chronic epilepsy. This is how the hypothesis is that the first hit is the radiation, or in human cases, it's actually genetically predisposed for epilepsy. The second hit is like something activates it. In human cases, it's like a fall or sleep deprivation, stress, all kinds of things can actually activate the seizures in these human subjects, and it can happen at any part of any stages of their life. So here we have exactly the same kind of model. So here we irradiated it, resulting in dyslamination, and then when you inject it with PTC, that is a GABA inhibitor, you get seizures in a moment. And when you actually hit the brain, that a slight brain injury can also cause these seizures. So what we did is actually as I said, we irradiated the mother, we isolate the neurons from the pups, and then we cultured the neurons for 15 days. So the neurons that are cultured for 3 days is considered to be like an adult, like it's equivalent to an adult. So I'm going to quickly go through the results, because I think it's running out of time. So here we have a protein. This is actually done. You can see the days of neurons in the culture, so 3 to all the way to 15, and you can actually see the growth of these neurons too. So this one is actually stained for a protein called GAF43, that is a growth associated protein 43 that is involved in growth axonal growth promoter, and then you have a greens that is FOMAP2, that is a marker for neurons. So you see that as the day passes, the XRT neurons has increased axonal growth, that increase. So that means they are hyperactive in growth. Actually, they grow a lot than the normal neurons. So if you see the typical neuronal analysis that you actually do for the dendritic, the Shawn analysis for the dendritic arborization, you actually see an increase in the dendritic pattern also for the XRT. The spines doesn't change much. And then if you actually go for another marker, that is the tau, that is an axonal marker, you see an increased complexity of the axonal structure here. This is on day 15. You can actually see the comparison between a normal neuron and an XRT. This is an excellent model in which we can actually take a complex animal model, split into smaller cell culture in vitro models, and then analyze the effect, and then go back to this animal model. So you see the path lengths are all different. So basically what this slide shows is that the XRT, that is the irradiated model, has increased axonal growth that can actually contribute to the factors that contribute to epilepsy. So here is another slide that shows actually these neurons have increased pre and the prosynaptic components in these neurons. So they have increased excitatory synapses. That is actually expected, because these neurons fire like anything. So that's basically what causes these seizures in these mouse models. And here you can actually see this is another marker for synapsin 1, that is a marker for matured synapses. So what it literally says is that like these neurons are actually talking to each other in a very efficient way. So all these red punctures here are matured synapses where these neurons communicate with so many other neurons. So this is a typical example of how, as I said, we can actually take a complex model organisms and put it into simple perspectives and see what changes that's happened. So again, this is another rescue study that we did. What if we can actually inhibit certain receptors, this would go back to the normal level. You have the baseline here. You can see in XRT this is like very much activated, and then you stimulate with glycine, that is an equivalent to the long-term potentiations in these animals, and you actually see an increase again. And then you block these receptors, glutamate receptors, it actually goes down in both these cases. This is how a typical example of how you actually use a disease model, and then you activate it further, and then you give a drug, and you see that the effect comes down back to the normal. So I think this is my last slide. So acknowledgement that this work won't be possible without my mentor here. All these trees, and thank you for your attention. Hello?
Hello.
Ah,
Thank you, Febin. Now the session is open for discussion. The participant can post your queries in the chat box. Thank you, Febin. Now the session is open for discussion. The participant can post your queries in the chat box. Thank you, Febin. Now the session is open for discussion. The participant can post your queries in the chat box. Thank you, Febin. Now the session is open for discussion. The participant can post. Febin, I have one doubt. What about
okay.
the treatment for this epileptic patients?
So the animal model that we use actually is cortical dysplasia. So there are many different treatments for these patients, but the model that we use is actually, we call it as a medically retractable disease. So they actually go through the treatment and their epilepsy doesn't get cured. That's why they come to the hospital for the surgery. So then only, so the first line of treatment for all these epilepsy is medication. So most of the people, nearly 40% of the patients, they won't respond to these medicine. So we don't know why they don't respond to this medicine. That's why they wanted to have the other mode of treatment is to surgically resect the brain samples from the. So I didn't actually touch that point. We actually do some kind of what do you call organotropic slice cultures from patients, human patients. But the problem is that the sample has to be young. Like if it's a young kid, the brains will survive more. Otherwise, the brain won't actually survive. I mean, the resected part of the brain. So all these experimental models are actually medically irretractable epilepsy. So they don't respond to medicine. That's why they come for all these treatment surgeries. So we don't know why they don't actually they don't respond to traditional medicine. So the other thing is that this is the cortical dysplasia usually happens in the cortex. It is not like the temporal lobe epilepsy there, in which you can actually very clearly see the lesion and you can take it out. Here, the brain MRI and everything will look exactly like the normal. So we have to put the in-depth electrodes inside to see in which area actually the neurons are firing, and the surgeon will actually take out that region. And once a surgeon gets the brain tissue from there, we will have to do a histochemical analysis, and then we see that, okay, the neurons are abnormal. Then only you will know that this is because of the cortical dysplasia. By the time it will be like the patient will be like around, he will be having epilepsy for the past, like, it dates from like 2 years to 20 years. So what happens is the surgery won't have an effect also, because epilepsy is that kind of a disease in which you get the seizure in one side and it actually passes to the entire brain, and then the focus of the region changes. So basically a normal brain in course of time changed to an epileptic brain. So yeah, the other thing is that we cannot actually shut down the entire brain just as we do it in the culture. If we cannot actually give all these blockers to the brain, then it will instantly kill the patient. So you have to be really tricky in what dose we have to give and how we have to give the patient these blockers. So that's why the animal model comes into play, in which you can use to what it fine tunes and see in what dose it is effective. But at the end of the day, you always have to work with the human to finally say for sure this is what is happening.
So can you see the question?
In the chat box.
How to make sure critical dysplasia is due to radiation and rat models and not from other factors working like environment, working like. How to make sure critical dysplasia is due to radiation and rat models. So I think the question she's asking is how to make sure the cortical dysplasia is due to radiation rat models and not from other environment factors working like environment. So the answer to the question is that we don't have a real control for this the irradiated model, because usually when you have a transgenic animal model, you will have wild type of, right? So it will be like always that you have a knocked down animal and you have a wild type also in the same litter. But in this case, actually, you irradiate the mother, so on an average, all the pups has the same effect. So we don't have a negative, a positive negative control for this. The only thing that we do is actually we keep one aside that is not irradiated, and we treat exactly like the same with the other one. So when you do the isolation of the cells, you exactly isolate it from both the animals, and you do it side by side. So you don't see that change in the non-irradiated wild type animals. So that says that it's not because of like working and it doesn't have an environmental effect in that.
Okay. Any further questions? Okay. Now I invite our research scholar, Reshma KR, to propose vote of thanks.
Respected and our most distinguished speaker of the day, Dr. Mohammed Febin Farooq, our beloved HOD, Dr. Emma LP, respected IQAC coordinator, Dr. Santosh T. Varghese, Danya Balakrishnan and Sharmila PS, assistant professors, Department of Zoology, Maharaja's College, and dear participants, a warm good evening. I deem it a great honor to propose the vote of thanks to all who have helped in making this webinar such a resounding success. I would like to thank our distinguished speaker, Dr. Mohammed Febin Farooq, for making such an excellent presentation and making this webinar highly informative. Thank you, sir. I would like to express our profound gratitude to our HOD, Dr. Emma LP, for her moral support and guidance in this webinar. I would also like to express my gratitude to IQAC coordinator, Dr. Santosh T. Varghese, for gracing this webinar with his presence. I would like to thank Danya Balakrishnan, Assistant Professor, Department of Zoology, Maharaja's College, for coordinating such an informative webinar. I would also like to thank Sharmila PS, Assistant Professor, Department of Zoology, Maharaja's College, for her presence. Finally, to all the wonderful participants who have turned up for this webinar, thank you so much for your cooperation. Once again, thank you all for your cordial cooperation. Thank you.